Okay, so thank you for a kind introduction. So I will be talk about synthesizing a minimal cell with artificial metabolic pathways. So recently, I moved to Kyushu University. So my biggest research motivation is what bridges the non-living simple molecular assembly and the living complicated or sophisticated forms of matter. So as you know, the living system is characterized such as by the presence of metabolisms or genes or reproduction behavior like this movie. So, but there's a big gap of complexity between the simple molecular assembly and complicated living system. So, so talking about the complexity of the system, so if we focus on the metabolism, complexity of metabolism, some researchers says that extremely simply speaking, such a metabolism is classified into three chemical units. For example, first one is the energy molecular production unit, that is the consuming glucose molecules to synthesize ATP, that is the energy currency of the living system. And consuming ATP, they synthesize or replicate DNA and synthesize the functional molecules, proteins. And with the help of proteins and using the byproduct of the glycolysis, they synthesize lipid molecules, and lipid molecules are incorporated into the cell membrane. Then the finally systems show the chemical reaction network, and they synthesize themselves. And then they realize the production. So in this study, so my research approach is constructing a transient system or intermediate system from simple molecular assembly to partially reproduce some characteristics of the living system by using a three chemical unit by the simple metabolism system. And this system is I called synthetic minimal cell system. Synthetic means I want to construct such a transient molecular system from simple molecular assembly by using simple and non-biological systems, because the biological system is already, sophisticated system. And such a system should be constructed in the steady flow of the inflow and outflow of the matter and energies. And with the couple of such a chemical reaction unit, the system also have to show such a reproduction cycle. That means the system have to show growth, and the membrane have to show deformation and division, and then the size recovery. So this is a reproduction cycle I called. So briefly, I will talk about the experimental setup to realize such a system from the simple molecular assembly. I applied the microinjection technique, like schematically shown here. This is toward the target vesicles. Food molecules or material molecules are supplied by the microinjection technique by this flow rate. So this is the brief summary of the chemical system, and I will from the next slide I will explain about this. So here the target vesicle is composed by the artificial member molecule, that is AOT molecules, which have a sulfonated head structures like this. And towards such a target AOT vesicles, the first food molecules, that is glucose molecules and dissolved oxygens, are supplied by microinjection technique. Here, with the help of enzyme, that is glucose oxidase, from the glucose, the supplied oxygen is reduced to the hydrogen peroxide. In the biological system, ATP is the energy currency molecules, but in my artificial simple system, the hydrogen peroxide is the energy molecules. And this energy molecule, hydrogen peroxide, is used to synthesize polymer. Here, the aniline molecule is similarly supplied to the target systems. Here, with the help of another enzyme, horseradish peroxidase, the aniline molecule is oxidized to the aniline radical cation, that is, activated form of monomers. And this radical cation molecule is highly reactionable molecules, and these activated aniline radical cations are polymerized on the vesicle surface to form the specific polymer called pan-ES. Here, the specificity of this system is the interaction between vesicle surface and the polymer. Here, the AOT vesicle, this is the picture of AOT vesicles, which have a sulfonated head structures, provide a specific reaction field for the polymerization of aniline molecules. And then in the presence of such a sulfonated vascular membrane, the polymerization of such aniline molecules are guided to form specific pi-conjugated polyaniline structure. This pi-conjugated specific polyaniline, which have a complex between the membrane molecules and polymer, this is called pan-IIS molecules. And the synthesis of such a specific polymer can be characterized such as by micro-Raman mapping by picking up a specific peak like here, or directly observing the unpaired electron by the electron spin resonance, or by absorption spectral measurements. And the interesting point is that when we replaced the vascular composing molecules from AOT to another molecules, for example, they are all sulfonated membrane molecules, AOT vascular membrane molecules, or SDBS surfactant molecules coupled with another, supporting membrane structures, or sulfatide molecules. These molecules are all, they can be all worked as template vascular for obtaining such a pan-IIS specific polyaniline structures. So in this sense, it's very, very simple system, but we can say that the composition of the vascular membrane can be partially reflected as the composition of the polymer structures like this. Okay, so finally, towards such a system, when we supply additional vascular forming molecule as a micellar solutions, these molecules are incorporated with the help of pan-IIS, which has the membrane molecules and polymer complex structures, and these supplied molecules are incorporated into the vascular membrane. Then I observed that the vascular membrane showed the radical membrane growth like this. This is the phase contrast microscopy images, and this is target AOT vascular membrane. Toward this vascular membrane, the food molecules are supplied by the microinjection technique, and the pan-IIS polymer is in situ synthesized on the vascular surface membrane. Then under such a steady flow and chemical reactions, the supplied AOT molecules are incorporated into the vascular membrane. Then the vascular membrane shows radical membrane growth like this. And compared to the control experimental system that do not synthesize pan-IIS system, the vascular growth coupled with the pan-IIS synthesis or coupled with the chemical reaction network show the exponential membrane growth like this. So the key point is that to construct such a vascular membrane growth system, the mutual catalytic relationships is important to design such a system. That means the target vasculars provide specific reaction field to guide to synthesize specific polymers, and the obtained polymer give a positive feedback towards the template vasculars to encourage the membrane growth. And this system is very much simple compared to the actual biological system. So we can make a list of the chemical reaction of this system, and we can write down the model reaction of this, and we can write down the rate equation, the rate of the chemical reaction of this system, and then we can describe the kinetics of the system. This equation still contains a priori undetermined parameters like red bold characters like this, but this is the artificial system, so we can make a control system when we omit the vascular membrane supply, and when we omit the sustainable food molecule supply. That means the polymerization in a closed system in a reaction tube like this. In such a system, the reaction begins in the first several seconds, and then the reaction reaches to the equilibrium. By using such a control system, we can determine such an undetermined parameter of the rate equations, and we can successfully describe such a membrane growth coupled with the artificial and very simple reaction network with no free parameters like this. So this is one of the advantages of such a simple molecular. This is the one of the advantage of the simple artificial researches. So toward the reproduction system, I talked about the how to say chemical reaction network and the vascular membrane growth. But the vascular membrane growth itself is not a reproduction. To construct a reproduction system, such a membrane growth should be guided to lead to deformation and spontaneous division and vascular size recovery. So I will talk about the reproduction cycle from now. So the deformation of the lipid membrane can be described by membrane elasticity theory, and this is the this is the spontaneous curvature model, one of the simplest membrane elasticity model. So this is the how to say phase diagram of the stable membrane shape. There is a two important parameter. First parameter is the volume surface ratio. That means the 1.0 means complete sphere, no deformation. But less than 1 means that there is an excess membrane area, and using excess membrane area, vesicle can deform. And the second important parameter is the spontaneous curvature of vascular membrane. So zero spontaneous curvature means, how to say, zero natural curvature. That means the, how to say, growth of the membrane or generation of excess membrane area lead to the this like a tube-like shape. And then according to this phase diagram, that to obtain the reproduction cycle spontaneously, the vascular membrane should have take this blue pathway. And to take this blue pathway, the membrane requires positive value of spontaneous curvature. Theoretically, such a curvature can be controlled by the molecular shape. This is the cylindrical membrane molecule features almost zero spontaneous curvature. And this is a picture of inverse cone-shaped membrane molecule features negative value of spontaneous curvature. And when we assemble these molecules to form bilayer membrane, by using only cylindrical membrane molecules, how to say, the zero spontaneous curvature membrane is preferable. But when we introduce second component, the inverse cone-shaped membrane molecules to this cylindrical membrane molecule like this, the redistribution of the inverse cone-shaped membrane molecules can, how to say, spontaneously generate the positive value of spontaneous curvature like this. So by introducing or by modulating membrane composition, theoretically, we may, how to say, control the deformation pathway of vascular membrane. And in my research, I applied the cholesterol molecule as a second amplifier. Previously, I showed the membrane growth coupled with the chemical reaction network, and in the previous pictures, the vesicle composition is only AOT, only the cylindrical membrane molecules. Toward this membrane, I introduced 10% of inverse cone-shaped molecule, that is cholesterol, and then we observed that the deformation pathway is successfully changed to the spontaneous neck formation and neck division. The only difference is the membrane composition, 10% inclusion of cholesterol molecules, and other experimental setup is same. The food molecule is similarly supplied to the target vesicle by microinjections, and PANI-ES is synthesized on the vesicle surface, and the vesicle membrane shows membrane growth by incorporation of externally supplied membrane molecules. But the deformation pathway is successfully changed, and the vesicle membrane shows the recursive membrane deformation and division like this. But the problem is that this system lacks the volume increase mechanism. That means the membrane surface area can increase by uptaking the additional membrane molecules, but you can see that the size of vesicle becomes smaller and smaller by deformation and division. So finally, I applied a volume recovery system by consuming the asymmetric permeation mechanism. Here, the fructose molecule and sucrose molecule has a different molecular size, and the membrane permeability of molecule is very much different. So in the initial state, when we encapsulate sucrose molecule inside the vesicle membrane, and the outside solution contains fructose solution. So even if the internal and external concentration is same, the asymmetric molecular permeation generate, how to say, the sustainable slight concentration difference between inside and outside of the vesicle membrane. This results the sustainable osmotic pressure differences and the volume inflation like this over the more than 10 minutes like this. So finally, integrating the chemical reaction network, molecular supply, and deformation pathway and volume recovery processes, I construct a synthetic minimal cell system I called. This is the A means normalized surface area, V means normalized volume, and according to the molecular supply, and the chemical reaction network, this initial mother vesicle show growth and division like this, and this left one, the left daughter vesicles again showed growth and division like this, and this grand daughter vesicles, the size of grand daughter vesicles have the smaller sizes compared to the initial mother vesicle membrane, but by introducing volume recovery processes, by waiting several seconds, these grand daughter vesicles recover their sizes to the almost equal to the initial mother vesicle like this. So this is the the reproduction that means growth, deformation, and division and volume recovery of the synthetic minimal cell system. I carried out 50 experiments by this. So here, the 45 initial mother vesicles showed first division, and then the 22, the initial mother vesicles showed another division, and then the 14, the grand daughter vesicles show another division like this. So sustainability become less and less, but I think anyway, based on such a strategy, I constructed the reproduction of vesicle system coupled with the chemical reaction network. So first short summary is that I'm interested in the emergence of living property from molecular assembly. And the first strategy is the mutual catalytic relationships that links each component, membrane and polymers. And here the vesicle surface guides pan-ES synthesis, and pan-ES promotes the uptake of amplifier, which results in the vesicle growth. And then the each linked component is after linking each component, the system shows growth coupled with the metabolic pathways, and such a growth system, growing system was guided to deform to vesicle division based on the membrane elasticity theory. So now the ongoing project is that here, in my previous study, the vesicle can successfully show the reproduction up to 4 to 5 generations. So the first ongoing project is sustainable reproduction. That means what causes death or what causes failure of the reproduction of the vesicle membrane. And another topic I quickly talked here is a framework of evolution. In my talk, the vesicle composition was AOT, and the polymer used was a polyaniline. And now I'm trying to include the SDBS, another sulfonated vesicle membrane, and polypyrrole. It's very similar, but these molecules are also synthesized with the template polymerization coupled with the vesicle membrane. And now by using vesicle dispersion system. Now, okay, I will quickly talk to before. I'm observing the very interesting catalytic network. This is the vesicle composition. This is the supplied amplifier system. And PANI or PPY means the catalytic polymer. Here, the growth of this is the supplied amplifier concentration, and this is the growth rate. The growth rate is nicely fitted by the Hill equation, and we can observe the various growth curve like this, and yeah, they are almost beautifully fitted by the Hill equation. So that means that by modulating the composition of vesicle membrane or polymer, we may construct a variation or selection or evolution by using this vesicle-based system. Okay, so the next project will be sustainability of reproduction or evolution of this self-reproducing system. So thank you for listening.
Thank you for the talk. Any questions or comments?
Hi, nice
Be
talk.
quick.
Maybe just a short comment. You mentioned about adding inverted cone lipids in order to make it a positive spontaneous curvature, but and then because of the redistribution across the leaflets, but the flipping of the lipids across the leaflets is very slow actually. So you might want to consider already making an asymmetric membrane, because otherwise I cannot see how it works. To call the redistribution, it will be very slow, I think.
Yeah, because quickly answering this question. So in my case, the additional membrane molecule are supplied externally by the microinjection technique, and the supplied molecule is first inserted to the outer layer of the vesicle membrane, and then transferred by flip-flop motion to the inner leaflet. So in this case, is such a first insertion into the outer monolayer, and the and inclusion of the first and second membrane component will be result in the positive value of spontaneous curvature. Can I answer?
Yeah, okay. I just think the flipping will be very slow, but we can discuss later. Yeah, thanks.
Quick question. Thanks for nice talk. In this division experiment, are you also supplying the cholesterol from the outside?
No.
Okay, so you eventually dilute the cholesterol concentration of cholesterol in the membrane, then so they at some point stop dividing, right?
Yeah, but here I have to say that the 10% cholesterol is the initial condition, but I change the value of the cholesterol concentration, and when the cholesterol concentration is reduced to only 1%, cholesterol inclusion still successfully result in the vesicle division. So I think the dilution of cholesterol is not so big a problem, I think.
Yeah, but if you eventually want to divide it
correctly,
forever, okay. Okay, thanks. Great talk.
Okay, thank the speaker again. Eight, 18 to 18.